It's my great pleasure today to introduce the ANZOS clinical keynote speaker, Professor Steven Smith. Professor Smith is the Scientific Director of the Translational Research Institute for Metabolism and Diabetes, which is a joint venture between Florida Hospital and the Sanford Burnham Prebys Medical Discovery Institute in Orlando, Florida. He is also the Chief Scientific Officer for Florida Hospital. Before this, he was actually at the Pennington Biomedical Research Centre, where he was the Director of Nutrition Obesity Research Centre, the Associate Director for Clinical Research, and Director of the Inpatient Unit. This is actually where I first met Stephen when I was a young postdoc at the Pennington, and he was an amazing inspiration and always available with words of wisdom and advice, and he's been actually instrumental in shaping not only my career but the career of many young clinical researchers. Dr. Smith's career in clinical investigation spans the translational spectrum from drug discovery, basic studies in cell culture systems, to clinical interventions in obesity and diabetes. His clinical work focuses on the early clinical development of novel pharmacotherapies and environmental approaches to weight management, and includes the role of energy expenditure and substrate switching in the pathogenesis and treatment of obesity and type 2 diabetes. Aside from his clinical studies, he's actually also well known for his extensive basic research with work on regulation of mitochondrial function using primary human cultures. To date, Dr. Smith has authored over 230 publications in top-tier journals, including Journal of Clinical Investigation, JAMA, Cell Metabolism, and Diabetes. He is also past president of the Obesity Society. Today, Dr. Smith will be speaking on unraveling the links between adiposity and metabolic disease. Please join me in welcoming Dr. Smith.
All right, Leonie, thank you for a wonderful introduction, brief, so we can get right to the science, which is where I always like to be. I do also appreciate the hospitality of Australia, of the Society, and of the wonderful time we've had here in Adelaide. And thanks to all of you for getting up, maybe having a little coffee and tea before or after your jog along the beautiful river. And it's a real pleasure to see you this morning. So when we were speaking on the phone, I guess it was almost a year ago now, talking about, what would I be able to offer to a very diverse group that has the theme of obesity and metabolic diseases, but also each of you have your own specific area of your expertise. And we came across this idea of an area that we've been working on for many years, actually, a couple of decades, which is posted here, and that's what is it about adiposity that leads to metabolic disease? And there are many different perspectives that one can bring to this question. And what I want to do is really acknowledge the multi-organ influences for how adiposity leads to metabolic diseases. The list continues to grow, obviously, with cancer being predominant and et cetera. But I'm today going to take an adipocentric view. I'm going to talk about adipose tissue as one of the key organs that changes in the disease that we know as obesity, and lessons from both basic and clinical areas that show us how adipose tissue does play a central role in the pathobiology. Now, I want to be clear on one point when I say that: a central role in the pathobiology of the development of metabolic complications of obesity and potentially cancer complications as well, although I'm not going to talk about that today. I do believe there was a nice session last night where we all raised our hand and tried to understand where the origins of obesity might lie. I'm not going to talk about that today. I'm going to talk about how adipose tissue actually contributes to the metabolic consequences. Okay, so let's go ahead and get started. As Leonie mentioned, I work at the Translational Research Institute for Metabolism and Diabetes, which is in Orlando, and our goal over every bit of work that we do is breakthrough treatments for metabolic diseases. Our themes are obesity and adipose tissue. We work also in type 1 and type 2 diabetes. We also have a strong exercise and aging group and just launched a NASH and NAFLD group as well. And we live adjacent to a strong bariatric surgery group that we work with carefully. Our approaches go all the way from bench to bedside and back. We do quite a bit of what I call deep phenotyping, and that is intensely studying small numbers of both patients and healthy volunteers as a means to understand how the systems work and how they become dysfunctional with the development of obesity and other metabolic diseases. I won't talk today, but we have also been working in the area of what I call quantitative bioenergetics. Oftentimes, we talk a lot about food intake, energy expenditure, the microbiome, what have you, and we begin to profile and pattern. And what we're trying to do in this area, or this approach, is really get quantitative down to the kcal level on how the checkbook of life actually ends up working. I will make disclosures here. I work in early target discovery and drug development in the clinic, and I will talk about none of these actually today, but I put them up just so that you understand my background. How many here have been to Orlando? Hey, probably seeing Mickey Mouse, Harry Potter, Shamu the whale. They live down in the bottom left-hand corner of the southwest part of Orlando. The rest of the city is actually wonderful, and I put a little map here, so if you're ever in Orlando and you want to come visit the center after you visit Harry Potter, please give me a ring. I'll put my email address up in a little bit, and I know I've already spoken with a couple of people that are going to make trips to visit us. So let's start with Steve Smith's perspective on the epidemiology of obesity and where we need to be spending our time clinically, and then I'll dive right into really the framework that we use to think about adiposity and how disease develops. This is an old slide, but there are multiple other newer references that show what's implied in this slide, and that is that throughout time, the histogram, the frequency of severe obesity has been increasing quite phenomenally. It's an interesting biological phenomenon, but also an environmental phenomenon. And we've actually gone back and looked at data on height and weight from the late 1800s, and you actually see a bell-shaped curve. It's a normal distribution in that environment and that age and time. And the reason that this becomes important is that from a clinical standpoint, when we look at the obesity epidemic in Western societies, certainly in the 80s, but absolutely at the turn of the century, we're beginning to see this push on the right-hand side of the frequency distribution into severe obesity, and that has huge public health implications for our health systems. And last night we had a little bit of a discussion around, is obesity a disease? And I kind of came, to that discussion a little bit more philosophically, but practically speaking, if you're in the healthcare business, which I would argue many, if not all of us are, that these severe obese patients are going to be a disease in and of themselves. And I think also in talking to insurers and other folks that on that side of the distribution, that there's less concern about, is this a disease, because we know the costs are high. And this is an example of where we sit in the United States, at least at this point in time in our projections. This is data from 2012, 5 years ago, but it's certainly true today. The numbers continue to track pretty closely that 10% of Americans will have a BMI over 40. So as you go out and talk to your healthcare systems, this is a factoid, if you will, that I think can bring people to their get people to pay attention to this, and can certainly help in making the argument that we have a real problem on our hands. 2030 being 10% of the U.S. population. Now, the other area, before I get into kind of the hardcore pathobiology, that I'd like to point out is that we're beginning to see people wake up to the impact of obesity in other traditionally non-obesity caring people. So the folks in type 1 diabetes, and I'm trained as a clinical endocrinologist, by the way, are beginning to wake up and understand that obesity is influencing not only the clinical care, but also the complications related to type 1 diabetes. And this is data from some of our colleagues around big U.S. database on the percentage of patients who have overweight or obese. And it's important to note that these kinds of interactions between obesity and other diseases are also going to become more important in our healthcare systems. We've seen that in orthopedics. I mentioned type 1 diabetes. You could go through the list, cancer again being one of the other hotspots. Okay. So on to the main part of the talk today. Our overarching premise is that you need a solid framework to understand causality, what leads to what, and to develop prevention and treatment strategies that improve health. And again, we're going to take an adipocentric view of the problem of obesity today and acknowledge that there are many other inputs and important areas that go along with it. So our framework really includes the amount, the location, and the location of where we store our fat is strongly influenced by hormones. I'll show you data in the second half that suggests that where we store our fat is epigenetically programmed. I'll talk a little bit about advances in our understanding of what a fat cell does and how it becomes dysfunctional and what functional actually means. And then we'll touch briefly on this idea of expandability, how the ability to grow fat cells and expand the adipose tissue is actually a healthy response to overconsumption. One note I won't go into very much today, and this is one that a friend of mine, Marianne Pellemounter, who was one of the primary folks who developed in the early days recombinant leptin, has been thinking about this for quite some time, is this question, is obesity like cancer? Is this expandability, the ability to make new fat cells and grow adipose tissue, akin to the ability of cancer cells to grow in an uncontrolled fashion? And the idea there is that adipose tissue may actually drive obesity in some people and pull energy into adipose tissue as opposed to adipose tissue receiving excess energy. And I'm not going to talk about that today, but I think there is some potential to actually consider that, particularly in the severely obese person. So what about the amount of adipose tissue? I think obesity in and of itself is visible. We walk around with it or we don't walk around with it. What I hope to compel you of today is it's a little bit more complicated than that, and that what we see on the outside sometimes is not indicative of what happens on the inside from a health perspective. And that is connected to this concept of metabolically healthy obesity. One note I would like to make in that whole area from, if you will, a little bit more philosophical perspective, is that the definitions by and large of metabolically healthy obesity only pertain to metabolism, diabetes. We don't know what it means to be healthy obese. Does that change your risk for cancer? We certainly know it has implications for osteoarthritis and joint diseases. So when we think about the constellation of the consequences of obesity, I think we have to be very careful that we don't narrow in too soon on what it means to have healthy obesity. So let's talk about location. We know going back into the 40s and some earlier reports that people had noticed that the shape of the body actually contributed to either the risk of developing disease or not. I point here to Jean Vague, who had a paper in 47 and this paper in 56, where he began to really lay out on paper for the first time, at least in the Western world, the relationship between apples and pears and disease risk. And we're going to spend a lot of time talking about that actually in the second half. But before we get to that, we're going to talk for a few minutes about different kinds of fat cells in different parts of the body. Now, everybody's aware of this concept of visceral versus subcutaneous fat in the belly and the harmful effects of large waist circumference. I would like to note that when we started doing CT scans now almost 25 years ago to measure visceral fat, we began to notice on our CT scans, if I don't have a pointer here, but if you'll look underneath the subcutaneous layer, you can see this white line that goes around all the way within the subcutaneous adipose tissue. You can see nerves and veins and arteries coursing through what we actually now know as a fibrous fascia. The surgeons have known about this for years, but I think many in the medical field had kind of missed this. I had missed this, many others have, and this was simultaneously rediscovered by our group and Brett Goodpaster. And let me just jump ahead here. This is on the left-hand side an ultrasound that was done in our clinic and another MRI scan where you can clearly see the delineation between a deep layer of adipose tissue and a superficial layer of adipose tissue. Well, why is this important? It turns out that this deep layer of adipose tissue, from a health perspective, is intermediate between the superficial or lower body adipose tissue and visceral adipose tissue. It has a completely different origin in terms of its embryology. It comes from a mesenchymal origin versus the superficial subcutaneous fat, which is an epidermal origin for all of you who've taken those kinds of embryology classes. It's an important distinction here from an anatomic perspective, but data that Brett and our group published some years ago and continuing work of a few people in this area highlight that this deep subcutaneous adipose tissue is metabolically unhealthy. We'll get to that in a minute. It's also interesting to note that the plastic surgeons who do a lot of liposuction, particularly large-scale liposuction, spend a lot of time working in under that fascia so that when they get the bleeding, it doesn't show underneath the skin and people think they're having an easier surgery at the end of all of that. And it turns out that we didn't really discover this. Sterzi, who was an anatomist in Italy back in the 1800s, actually has a whole volume on the quality and differences in adipose tissue throughout the body. And he termed this demarcation between a deep and superficial adipose tissue as the fascia superficialis. And again, the surgeons will know it with different names in different parts of the anatomy. Now, the key components here are that this deep subcutaneous adipose tissue contributes to all of those things we measure in the clinic: your weight, your fatness, your waist to hip ratio, your DEXA measures, and that sort of thing. But it's largely ignored in the literature about how adipose tissue leads to the development of metabolic complications. So we're learning more about the quality of this adipose tissue, and importantly, it is highly correlated with the amount of visceral adipose tissue. So for those of us who work in the clinic, how many of you have seen what we affectionately call the olive with 4 toothpicks? Okay, the olive with four toothpicks. It turns out that those individuals in the clinic have lots of deep subcutaneous fat and a very thin rim of superficial subcutaneous fat, and very low amounts of fat below the waist and in the arms, hence the 4 toothpicks. And there is this component of, if you will, lipodystrophy or atrophy of the superficial layer. Alternatively, one can say that maybe there is an inability to grow that superficial subcutaneous adipose tissue, an alternate view. Now, moving on to a different and almost forgotten adipose tissue depot is the marble in your steak, the intramuscular adipose tissue, which was first described several years ago by Brett Goodpaster and Lauren Sparks, who's shown in the a photograph here in our group, has been actively working on the characteristics of intramuscular adipose tissue and has some really fascinating new work that I hope at some point you'll be able to hear about. But it's when you add it up, this, particularly in elderly folks, there's as much intramuscular adipose tissue in some people as there is visceral adipose tissue. And it has direct influences on skeletal muscle metabolism and characteristics that are completely different from subcutaneous or visceral adipose tissue. So what I'm trying to give you here is a little bit of a survey beyond brown fat, which I'm not going to talk about today, where all of these different adipose tissue depots are having their own independent effect on metabolism because their characteristics, their origins, and their functions turn out to be extremely different. So let's shift gears a little bit and talk about quality and function of adipose tissue and provide hopefully a little bit of a framework around that. So what we have known for many, many years is fat cells come in different sizes. And many investigators back in the 60s and early 70s set out to describe how the differences in adipocyte size related to the development of disease. On the top left is a hyperplastic adipose tissue where the fat cells are big, but they're not huge. On B are the hypertrophic obese patients who have really huge adipocytes. And in fact, the field is a little bit stymied because these fat cells get so big they're hard to isolate and actually study because they're very fragile around the edges oftentimes. And I think we have, let me just see here, we have a little black box there, but suffice it to say that these adipocytes in different sizes contribute to the development of metabolic complications. And this is one of my favorite slides. On the y-axis is insulin sensitivity. On the x-axis is the size of the fat cell measured from an abdominal biopsy. And this is Chris Wyer's work from 2000, but there are many, many studies showing the same kind of effect. And I haven't, at least in our North American meetings of the Obesity Society, it's very uncommon to talk about hyperplastic versus hypertrophic obesity. And we know from this and other data that the metabolic complications are very tightly linked to the size of the fat cell. So for the basic scientists in the room, how does a fat cell get big? Is it just overstuffed, or are there control systems that say when a fat cell gets so big, make a new fat cell? And indeed, there's evidence for that both in humans with pharmacologic intervention and in rodent models that we can continue to make new fat cells throughout the life. Therefore, if we continue to expand adipose tissue, that we may actually be healthy from a metabolic standpoint. So just giving you some of the ideas about how we think about adipose tissue. Now, the other thing is when we talk about metabolic diseases is everybody seems to be fixated on insulin as a glucose hormone. But insulin has pleiotropic effects. It's a mitogen that replicates new cells under certain circumstances. We've been focused on how insulin regulates lipolysis and stimulates lipogenesis, the synthesis and storage of fatty acids. But there's also work that shows that insulin has dominant vascular effects that can change how we think about blood flow within different organs, and adipose tissue is one of those that we've worked on around this same concept. Now, this is unpublished data from a body of work that we did about 5 or 6 years ago, where we looked at what happens to free fatty acids in the blood after you give insulin during euglycemic hyperinsulinemic clamp. And there's this very interesting relationship between insulin sensitivity and free fatty acids in the ability to have insulin turn off lipolysis and turn on lipid uptake and storage within the adipose tissue. And this is a key function of insulin sensitivity that is largely adipocyte-driven, that when we hear the words insulin sensitivity, we always think glucose. Begin to think about insulin sensitivity for the adipocyte. And we know that as body weight goes up, insulin sensitivity of the adipocyte goes down as well. And this is tightly linked with the size of the fat cells. So small fat cells are known to be insulin sensitive. They turn off lipolysis and they can store fat and take up glucose very rapidly. And so when we think about how adipose tissue leads to metabolic disturbances, don't just think about glucose. Begin to think about fat metabolism. It's a little bit of a challenge to those of us who grew up in the endocrine clinic using insulin to treat diabetes, but it's incredibly important to consider. So the other model besides hypertrophy and expandability is an endocrine model, and I'm not going to talk too much about this today. Suffice it to say that there are now an entire alphabet soup of adipokines. If you'll notice, I didn't put very many of them on here. And if you study those adipokines, don't take that the wrong way. I think I put here the ones that have, at least today in my mind, a preponderance of evidence that say that they are clinically relevant and that we understand their biology sufficiently to put them on the list. Now, that said, the soup of cytokines and the inflammatory milieu coming out of adipose tissue continues to be better understood, not just from the macrophage standpoint, but the other T and B cells and other cells that live within the adipose tissue. And we're also learning that there are bioactive lipids that are coming out of the adipose tissue that are exerting effects potentially in different organs underneath the umbrella of an endocrine model. All right, shifting gears a little bit, what else do we know about the function and quality of adipose tissue and how it relates to metabolic health? So on the left-hand side here, let me walk you through two photomicrographs that were stained for either the plasma lemma, which is the green circle around fat cells, or actually the capillaries that we can stain in the adipose tissue, which is red here. And if you look in the top slide, this is a lean, healthy, adipose tissue photomicrograph, and you can see lots of capillaries. Now, this is the extreme on one end, and on the other side, if you look below at the obese patient, you can see probably 4 or 5 little red dots here in the photomicrograph. Now, this loss of capillary density has been described in other tissues, and the phrase that's used to name this is called rarefaction. And the folks in hypertension understand rarefaction very well. And Magda Pasarico, when she was in my laboratory, and others that you see here, did some very nice work where we actually measured not just the capillary density, but we measured the oxygen tension within adipose tissue of lean and obese patients. And as you can see here, as body fatness increases, both the capillary density and the oxygenation go down within the adipose tissue. This is not the degree of hypoxia that you see classically that activates HIF-1 alpha from a signaling standpoint. It's between normoxia and true activated hypoxia, but it has profound influence on the metabolism of fat cells. We know that from animal studies. Ours were the first clinical studies, but there have been other studies now that highlight the human adipocyte is exquisitely sensitive, as other tissues are, to lowering the oxygen tension. Now, for our surgical colleagues in the room, this has been described in other adipose tissue areas and is in that literature related to wound healing and the ability to recover after a surgical insult in terms of simply being able to get the tissue and the cutaneous tissue even to come together and heal up. This has not been looked at in the surgery space in terms of how capillary density influences infection risk. And if anybody's interested, send me a note at my email later, and I can send you some of the surgical literature that goes along with that. Now, with regards to what this microhypoxia does to tissue, it's a very interesting concept to me, at least, and I still don't understand this to this today. But as body fat goes up, VEGF, which is supposed to stimulate angiogenesis, actually goes down, and this has been seen in rodent models as well. So you get this tissue that doesn't really seem to have the capacity to make new capillaries, and there are quite a few different gene regions and sets that go along with this microhypoxia. And in fact, the next one to talk about is on the bottom right-hand panels here shown as H, and that is as oxygen tension goes down, the development of fibrosis goes up. And fibrosis is really the next way that adipose tissue becomes dysfunctional. Our paper in 2009 showed this in humans. There was one paper in 2008 in rodents, and now an entire field of study around how adipose tissue becomes fibrotic during the obesity process, and probably serves to limit the growth of adipose tissue in the same way that not having enough capillaries will limit the growth of adipose tissue. In essence, taking that idea of expandability and saying you don't want to have adipose tissue expand forever, we don't have our necks grow this big, other than in some lipodystrophic states. We have fat under the scalp, but it knows when to stop growing. And so we think combined here that these processes limit growth of adipose tissue in certain areas. And then when they're absent, we may actually grow adipose tissue in other areas. Think of our arms, think of our hands, think of other areas as well. So these control systems for expandability are probably related to angiogenesis. They're also related to the development of fibrosis. And even we're now understanding that fibrotic adipose tissue is associated with fibrosis in the liver. So Corinne Clement in Paris has some really cool data on how the amount of fibrosis in visceral adipose tissue is related to the steatosis and, importantly, the fibrosis that occurs in NASH. Okay, so before we switch over into a little bit of hardcore cellular molecular biology, just to recap here, our framework for understanding adipose tissue and particularly metabolic disease is how much you have, the location, and I haven't talked really about hormonal influences. That's a huge component. Next, we'll talk about epigenetic programming. I hope I've compelled you that dysfunction of adipose tissue in some fairly unique ways is influencing metabolic disease. And then lastly, what we see on the outside, the total amount of adipose tissue really does depend not just on the food intake, but it depends upon the ability of the tissue to expand over time. Okay, on to part two. And Russell, if you could switch over to the second deck. We're going to go very cell and molecular for all of you basic biology gearheads. Fantastic. All right. I love these cartoons. Anything that has anything to do with obesity that shows up in a magazine, I clip it and save it. This was one recently. It's mostly just excess food turned into fat weight. Now, the young yoga lady on the left who's stretching there, I can promise you that being young and female with what appears to be a pear shape, you could feed her for probably a year and she would not develop the central adiposity that her colleague to her left has, okay? And the question then becomes, I've shown you how adipose tissue exists in different places. When you overeat, what actually controls where you store that fat? It's a very fundamental question in human and other biology that has yet to be answered. A parallel question is, why do women have more body fat than men? Does anybody in the audience know the answer to that? So that is what the teleological purpose is of more body fat. But how? What is the mechanism by which? Okay, estrogen. We're getting somewhere. This is good. So estrogen, as I said before, testosterone, growth hormone, cortisol, all have strong influences on fat patterning. But let me go one more layer. How does a fat cell in one part of the body know to respond to those hormones in a different fashion? It's a very fundamental question in receptors. Okay, well, we're going to go one layer deeper than that. Thank you. Yeah, this is wonderful. Because see, when you begin to ask what one of my pilot friends calls the 5 whys, you get to some very interesting places in science. And this was a 5 why beginning to this project. We had a little think tank. Some people paid us to go into a room every quarter for 2 years and think about the fundamental question, why are women fatter than men, generally speaking? And why does fat get stored in different places? So this second half is really a 10-year journey that we started, and we're beginning to get insights into at least how fat cells in different parts of the body are controlled. I won't talk about male-female today. I'm going to talk mostly about upper and lower body fat, and here's why. So again, going back to Vogue in the 40s and work that was done in the 60s and 70s, we can be apples or pears, okay? And by and large, men only come in one shape. If you're in the clinic, I promise you, if you ever see a man who is a pear shape, I want you to pick up the phone and call me, okay? But women, by and large, have quite a range of difference between being gynoid or android or upper or lower body fat. And that has a profound influence on the risk for developing metabolic disease, and I'll show you in a minute. Now, what we're also learning, and it's exemplified in this next slide here, is that cardiovascular risk is not just the absence of abdominal fat, either visceral, subcutaneous, or deep subcutaneous, but there's a protective effect of lower body fat. And that's shown really on the right-hand side. There's an independent protective effect for cardiac events in multiple studies. And this, importantly, is true in both women and it's true in men. So we often think about apple versus pear as being a female issue, but it also turns out to be a male issue as well. Not going to spend a lot of time on that today, but the epidemiology is very clear. We know that these olives on a toothpick, all right, that I was talking about earlier, because of the lower body fat, are at much greater risk than men who have equal amount of truncal fat but have lower body fat simultaneously. So what do we know about the function of fat cells from different parts of the body? Through the years, going back into the late 70s and through the 80s, I put some prototypical work that was done by Susan Freed, one of my collaborators, and groups in New York and groups in Europe as well, that highlighted that if you take tissue from a biopsy, study it in the Petri dish, there are a wide variety of different functions that we can understand about the tissue. And in contrast to that paper I showed you earlier where big fat cells lead to insulin resistance, that was abdominal fat. Turns out that lower body fat cell size has no relationship to insulin sensitivity. And in fact, lower body fat under healthy conditions, the fat cells get really big. And so we're going to have to rethink what we define as being a healthy fat cell based upon size, but clearly abdominal fat, big fat cells are bad. And this is an interesting study, I hope, for the clinicians in the room to understand how lower body fat contributes to metabolic health. These were women who were studied pre and post high volume lower body liposuction. And they were coming in for cosmetic purposes, but these folks decided they would do a study of their metabolism. And as you can see on the left-hand side, they had a pretty big change in their leg fat mass just from, again, multi-kilo lower body liposuction. And on the right-hand side, you can see that postprandial lipemia got much worse when they took all these fat cells out. So it really depends where your fat cell comes from, what the influence is going to be on metabolism. And this is really the converse of how lower body fat can be protected. So again, the framework and how we think about this, lower body fat can serve as a metabolic sink. We know that it's very insulin sensitive. It can expand quite easily. Doesn't make some people happy, but it's metabolically healthy, okay? We also have been working under parallel endocrine hypothesis that lower body fat may secrete a beneficial factor, or it does not secrete a deleterious factor, and we have ongoing work in our laboratory on these two ideas, which we'll get to in just a few minutes. And if you're interested in how sex differences contribute to the biology of a pear shape, there's a nice review that we published a few years ago in 2012. Calypso did a really nice job summarizing the literature up until that point. It's a really good read. So that leads really to the core molecular and cellular aspects of what makes a lower body fat cell different from an abdominal subcutaneous fat cell. So we, along with my colleague Susan Freed, who's now at Mount Sinai, started a little over 10 years ago a search for depot-specific signatures, and we started with the global hypothesis that there might be epigenetic programming, which I'll define here in just a few minutes, that there was an epigenetically different program in a fat cell that made it respond to hormones differently and exerted characteristics that allowed it to be cardioprotective. And as you can see here, DNA is very complicated. It's very big and large, and it wraps on itself to form chromosomes, but there are also exquisite control systems that determine which gene regions are actually open and available to be turned on, and which gene regions are actually closed. Now, that epigenetic marking can occur through DNA methylation, or it can occur through histone marks, shown here in this slide as epigenetic factors. And the chromatin then structure we can now interrogate in many different ways to really figure out what's going on at the level of DNA in terms of its epigenetic programming. How many people in the room work on maternal fetal programming or early life programming? We have a few in the room. You know, this concept that epigenetics controls metabolism, aka the Barker hypothesis, is one that's been around for quite some time, but the tools haven't really been available to be able to interrogate this at the molecular level. And these new tools, as I'll show you in a few minutes, are quite powerful for being able to pick out differences in epigenetic programming in multiple tissues and organs. So we went out just to survey and see what the mRNAs, microRNAs, long non-coding RNAs, which we'll get to, how they might differ between these tissues. So we just did simple liposuction in the clinic for either abdominal, and our gluteofemoral biopsies, by the way, are really on the lateral thigh. So gluteal, femoral, we take all of our biopsies in this region. Gluteal actually turns out to be different than what's below, and we actually think that the thigh or this gluteofemoral area is really important for the disease process. Okay, so on the left-hand side is the liposuction material that we collect. We digest it, we spin it, the fat cells float. The stromovascular fraction, which are the preadipocytes, are down in the bottom, and we can then purify the preadipocytes and study them in the dish. Now, in the next couple of slides here, I'm going to show you not exactly how we culture these cells in the dish, but I'm going to show you that many of the signatures that we see within upper versus lower body fat are preserved in the tissue after it's been cultured in the dish. And that is a very important component that we'll get to here. So again, we take these biopsies from both men and women and process them. And this schematic I'll use throughout, so you will have a little code for what we're actually measuring and studying, hoping it makes it a little bit easier. So our first experiment was in the tissue, where we did gene chip analysis. And we began to see really small sets of genes that were different between these different fat cells. Some of these in the Venn diagram in the middle were common between men and women, and those were the ones we really keyed in on, because we believed that potentially the ones that were not common were driven by sex steroids and other hormonal effects. And quite quickly we started looking at the gene lists, and we honed in on a category of gene called the Hox genes. Now, when I went through and studied undergraduate biology, I don't think Hox genes were discovered yet, and I'll walk through with you what is a Hox genes, because what we're seeing here is that we have patterns of genes that are in these Hox gene clusters that differentiate what a gluteal or lower body fat cell is from an upper body fat cell. As I move forward, I want you to pay attention to two of them, Hox A5 and Hox A10. You can see A10 is turned on in gluteal tissue, and A5 is turned off in gluteal tissue and up in abdominal tissue. Now, what does that mean? Well, Hox genes all come in a cluster. They live together. They are the genes that, by and large, determine how our body is constructed at the embryological stages, and they are the result of an ancient gene duplication that happened many years ago. Don't miss out this MIR196B. All of these Hox clusters have mirs, or microRNAs, and they all have what are called hot air genes, which are long non-coding RNAs, which I'll cover in detail in just a few minutes. But suffice it to say, we're seeing patterns of Hox genes in this Hox A cluster that look like they serve as a fingerprint for where the fat cell comes from. Okay? So there we go. The Hox gene clusters are 4, A, B, C, and D. They are on different chromosomes. We're going to talk about chromosome 12, a little bit about chromosome 7. And if you'd like to read about them, I put a few references here. The one in Nature Reviews in Cancer in 2010 in May is just outstanding as well. Now, earlier studies, once we did this work, we went back and looked, found that fibroblasts, not adipocytes, but fibroblasts in different parts of the body have their own barcode for where they come from, and it's determined by these Hox gene clusters. And basically, they were able to take a fibroblast from anywhere in the body and just based on the Hox genes tell you where it came from. And that pattern of Hox genes we think is also important, at least in superficial subcutaneous adipose tissue, but there is an established literature here that this barcoding is indeed important. So let's think about the cells that come from that tissue. We know that hormones can influence the gene expression within tissue, and the microenvironment's critically important, but what about the cells that come out of there? And this is work that was done by a postdoc in the lab, Adeline Devou, who cultured these cells over time, as I showed you. And if you look in the top two panels at Hox A5, or one of our friends, and Hox B8, in the dish, outside and independent of nutrition, environment, and hormones, Hox A5 is only turned on in pre-adipocytes that come from the abdominal depot. And on the converse side, take a look at Hox A10 and Hox C13, which have the flip side of gene expression. So we can now, very simply, with just two genes, Hox A5 and Hox C13, tell you if a fat cell came from the upper or lower part of the body. And this becomes important in a minute when we talk about epigenetic programming. So let's move on to going back to the tissue, where we then went through and profiled microRNAs. MicroRNAs are about 21 nucleotides long. They control other RNAs. And think about them as the volume on your stereo. They don't change the channel, but they just change the volume a little bit for many, many different genes. And we also found microRNAs that were uniquely expressed in upper and lower body fat. On the top panel here, I'll show you two, MIR203 and MIR3545, that are only turned on, for example, in male lower body fat. Now, functionally, we don't know what these are doing, but we do know, for example, that this family of MIR196 genes, which are in each of these Hox clusters, are only, again, turned up in gluteal adipose tissue, and they only differ, these MIR196s, by one nucleotide. So they're very highly conserved, and they seem to be regulating gene expression in a local fashion, in a gluteal specific fashion. And Adeline's gone through and defined some gene regulatory networks where she's beginning to define what it is to be uniquely a gluteal or protective versus an abdominal or non-protective adipose tissue, and went through and overexpressed MIR196 and showed that these target genes downstream of MIR196 in human adipose tissue can be downregulated by MIR196. And so what that means, again, going back to the earlier data here, is that in gluteal tissue, these MIR196 genes in these Hox gene clusters are actually turning off downstream genes now that we have this functional data to support that. All right, who's heard of a long non-coding RNA? We have some gene geeks in the family. I'm trying to see here okay, good. I'm glad I put this slide in then. So what is a long non-coding RNA? So we talked about microRNAs, which are 21 base pairs long. They're little tiny micro things. These are long, right? They're very long. They're non-coding. In other words, they don't make a protein, so they don't code for a protein. And for a long time, people thought these were junk RNA floating around, that they were getting in the way of doing good science. But what we now know is that these long non-coding RNAs act like transcription factors. That's shown in D, in C, and in A. And what they do is they bring proteins together on the genome. They sit down on the DNA, and then they can serve as either enhancers or they can inhibit gene transcription. So who's heard of PPAR gamma? PPAR gamma, we have drugs for PPAR gamma. Think of this like PPAR gamma with a ligand, and the ligand here is the long non-coding RNA. They exert powerful effects in cells to control gene transcription in the same way that thiazolidine diones have powerful effects in the clinic to regulate PPAR gamma-related genes. So, of course, we went through and profiled these long non-coding RNAs, and there's one in here that caught our attention very quickly, and it's called hot air, and it lives right next to this Hox gene called C13. And was commonly differentially expressed between upper and lower body fat. Now, hot air on the left side, on the left side is our gene hit list. On the right-hand side is showing you that it's only expressed in gluteal adipose tissue. We actually almost missed this from a bioinformatics standpoint, but it's clear. It's a beautiful signature C13 and hot air both. And they live in this chromosome 12 cluster that we've been intensely studying now for the last 4 or 5 years. So let's go into the tissue and the cells, talk a little bit about hot air. Again, hot air is only expressed in lower body fat cells. We think is uniquely involved in programming those fat cells. And when we look in cultured cells, it's cell autonomous, so it's not strictly being driven by the hormonal or microenvironment of the adipocyte. So we went back to the laboratory. We took fat cells in the Petri dish, shown in the top here, and we overexpressed hot air in abdominal cells, because, right, they don't make hot air. Lower body fat cells do. So we were thinking, couldn't we take an abdominal fat cell and make it healthy and look like a gluteal fat cell? And this was the beginning of that set of experiments. And if you look in the bottom two panels, when we overexpress hot air, we get two things. One is the fat cells differentiate into fat cells. The preadipocytes differentiate into fat cells. And then secondly, each fat cell gets a little bit bigger. Does that remind you of lower body fat, where we know the fat cells are bigger but healthier, and they have this high ability to take up lipids and sugar? And so this data, which was published a couple of years ago, was interesting to us. But this was the kicker. This is data that was produced by the GIANT Consortium, looking at genes that control body fat patterning defined by the waist to hip ratio. And if you'll look in the yellow dots in the bottom left-hand corner here, right underneath that, you can see that this Hox gene cluster on chromosome 12 had the strongest genetic influence on whether a person has upper or lower body fat. So this was a bit of a eureka moment when our clinical genetics and our fundamental cell biology were coming together to really tell us that these gene regions and genes that we were working on were going to be incredibly important for understanding metabolic diseases. Okay, let's see if we can go forward. Russell, I'm having trouble going forward. Could you maybe give me... Oh, this is good. So what does this have to do with epigenetics? In the last little section here, maybe another 5 minutes. If I have 5 minutes? Okay, good. So what is the evidence for epigenetic programming underlying these special lower body fat cells? Again, going back to our global hypothesis that this is epigenetically driven, taking the tissue and pulling the stem cells, the preadipocytes, out of that tissue, we then went through and began to think deeply around hot air and how it might actually work. Now, fortunately for us, hot air has been studied in, believe it or not, breast cancer. It turns out, at least in breast cancer, it is estrogen driven. That'll make our clinical folks very happy to hear that. And we wanted to try to understand in a fat cell what are the marks at the histone level that would give us some idea of how hot air was actually changing the other gene programs within there. And we focused in on a couple of these histone modifications. The little red dot there is a repressor mark, H3K27 trimethyl, and the H3K4 dimethyl is an active mark. So when genes are turned on, they can have one or the other of these marks. Okay, and I'll show you data that come back to this. The active mark is green. I hopefully color-coded this for simplicity. The red mark is actually going to end up in being off. So we started looking at these cultured cells again in the dish, and we did a very simple experiment with HOXA5, our old friend, that is turned on, as you can see in the left panel, in abdominal tissue. And when we do a technique called CHIP PCR, we can interrogate this gene promoter, and as you can see, the green mark goes along with the expression of HOXA5, and the repressive mark is on in gluteal fat, again suggesting that we have tissue differential expression. Going back to hot air actually looks like it's controlled by the absence of a repressive mark. You have to get rid of that. Now, this is my last slide, and then I'll conclude here. We have then moved that simple set of experiments into genome-wide scanning using both chromatin immunoprecipitation, or ChIP-seq, but also a new technique called ATAC-seq, where you can actually tell whether the chromatin is open or not. Now, in this setting, you have our two favorite genes here, HOXA5, which again is turned on in abdominal adipose tissue, and hot air, which is actually turned off in abdominal but turned on in gluteal. And the different histone marks here, I don't have a pointer on them, but I think you can see the two genes look very different in terms of what their histone marks and the degree of open chromatin looks like here. And so we're now going through this list of understanding what makes a gluteal fat cell different than an abdominal fat cell, and what are the genes that determine then the phenotype of that cell moving forward. And more importantly, we now know, and I don't have all of the data to show you today, didn't have time to do that, but we now know that individual variation in these genes correlates with waist to hip ratio. So they're not only determining the difference between boys and girls, but the variation in these epigenetic marks is determining the degree to which one is an apple or a pear. So I hope today I've given you a framework for understanding adipose tissue, disease, and these are the areas that we have, I think, to talk about in the Q&A session. So thank you, Leonie, and thank you to all of my collaborators on this second half of the talk.
Thank you, Steve, for taking us through that fascinating world of adipose tissue. We've got time for a couple of quick questions. If anyone, if people would like to move down to the microphones, just while I guess everyone's having a little think, I'm just wondering, how then do you suggest we go from, I guess, having that dysfunctional adipose tissue to the functional adipose tissue?
Great question.
Yeah.
Great question. So we know by and large that there's a little bit of a ratchet here, that when you have dysfunctional adipose tissue, that there are some reversible and there are some non-reversible aspects of adipose tissue function. Take fibrosis, for example, okay? That fibrosis, once it begins to pile up, has implications for how the cell signaling works and other things. And you can shrink the lipid within the cell, but the fibrosis really turns over very slow. So that can be, I think, a challenge. I think what we should be doing is trying to intervene before we get that full-blown fibrotic, hypoxic, inflammatory adipose tissue, particularly in the abdominal adipose tissue. And prevention of, and this is where the clinical piece is, prevention of weight gain in people over time, I think for that reason, is as important as for preventing diabetes. I think they probably go together. So some of these defects like adiponectin and some of the inflammatory components can be reversed. Some of them are going to end up being permanent.
Yeah, that was an amazing talk. Thank you. What sort of, Tanya Markovic, what sort of, from Sydney, what sort of epigenetic findings have you made? What things happening in what sort of things are changing these? What are the actual epigenetic changes that are happening that are driving this?
Yeah, so your question is what's upstream of all of this?
Yeah.
A couple things. We know, I mentioned that in breast cancer, for example, that hot air is estrogen controlled. We don't see that in adipose tissue. We think there may be other genes that are hormonally controlled that then impact this control point. For example, we had a paper a couple of years ago around growth hormone, which is another really good repartitioning agent that can reduce abdominal fat. Those growth hormone receptors are under epigenetic control, and they're only turned on in abdominal fat cells. Okay, so we're beginning to learn how the epigenetic programming of each fat cell determines the responsiveness. We think actually, and many others do as well, is that both male and female factors, both preconception and in utero, are going to determine what lays down these different marks. We know from longitudinal clinical studies that nutritional stress during pregnancy predisposes to higher waist circumference, higher lower gluteal amounts of fat. So we think that most of this will be programmed in utero, but then modified by hormones. And what that exact in utero event is, that's for others to work on, but we have some ideas.
Okay. Just one more thing, the
Yes.
fibrosis issue is very interesting, and it doesn't, of course, as you would know, it's not only in the abdominal fat, it's everywhere.
Yes.
And people who are great. You didn't talk about it a lot, but I think it's very interesting, the people who can expand, who are quite different.
Yeah. Yeah,
who look amazing, you know, and they also have all that fibrosis that's quite inflammatory, and they get the terrible PANIS.
That's right.
And all the rest.
So I would differentiate, and I didn't have time to go into this, between micro and macro fibrosis. Right. So the fibrosis that is probably most important for the fat cell is the microfibrosis that sits around the adipocyte. The big bands of collagen, which we know in our hands from our biopsies and surgeries, et cetera, we don't think that those are really contributing to metabolic disease, because they're just tension-related fibrosis. It's the pericellular fibrosis, which is going to be the real bad actor, that goes along with inflammation.
That makes sense.
Gary. Steve, hi, Gary Leong, University of Sydney. Magnificent talk. While I was listening to you, I was thinking, you know, some of these drugs like HDAC inhibitors are now in the cancer clinic.
Yep.
You know, they may be like a big stick on to try to modulate some of these microRNAs and things, but is there any data from those patients about fat distribution, and can you foresee, or there are drug companies now developing targets for these?
Yeah, great question. So first off, we do have an HDAC program within our adipocyte biology group, and so we're very keen on that concept. Secondly, from a clinical perspective, I sent one of my colleagues over to a metabolism and cancer talk at the Keystone meeting last year. There was only one talk about anything clinical at that meeting. So they're working, you know, metabolism and cancer people are working at the level of the tumor, and they're not beginning to think about how these drugs are actually influencing the rest of the body. So I think that is ripe for clinical investigation. There's a lot of work that needs to be done there, and there's certain HDACs that actually we're now learning are controlling different adipocyte phenotypes. So for example, beige versus brown. We think that we have our eye on an HDAC for upper versus lower. I mean, there's a lot of work to do here in the epigenetics, and this is going to explode because of the cancer people into the future. But great question.
One quick one.
Yeah. Steve, Nick Holmes, is there any work looking at transplanted fat in different regions and what happens?
Humans or rodents?
Any.
Yeah, so we saw some nice work yesterday in one of the sessions for being able to transplant human fat into rodents, and there are a variety of groups that are doing that as well. And I think one of the key experiments that somebody needs to do, I hope it's not us and somebody else will take it on, is to take these upper and lower body fat cells and transplant them into mice and look at their metabolic effects. I think that would provide us with additional ammunition that there might actually be a protective factor coming out of lower body fat. But great question.